Hello and welcome to the Hygiene Hangout, the hashtag No Filter Hygiene podcast. We hope you all had a great Christmas and New Year's break and enjoyed our last episode as well. Knowing that Daddy had a direct line to the North Pole certainly made my house a very happy and compliant one on the run up to the big day. But it's back to business in 2025 and that's exactly what we plan to do today. We have a very technically focused episode for you where we're going to be discussing environmental monitoring programs, or EMP as it's more commonly known within the industry. Today's special guest is someone that's been present within my journey in the food industry for well over 20 years, and he's been there to provide advice and support as not only a colleague but as a friend as well. I'm sure you wouldn't mind me saying that he is the Yoda to my Luke Skywalker. So sit back, relax and enjoy episode 7, Insight EMP+. Yeah. So Peter, welcome to the Hygiene Hangout. First things first, and for anyone who's been living under a rock for the last 38 years, could you please introduce yourself, tell us a little bit about yourself, who you are and what you do?
Yeah, Pete Littleton. I'm UK Technical Director for Christian's Food Hygiene. As you say, this year marks my 38th year in the food industry. Food safety the whole way through, from starting as a microbiologist in a cannery, right the way through, including hygiene, technical, production management roles, but for the last 25 years in food safety support with a variety of chemical companies.
Brilliant. Vastly experienced. Had a huge influence on my career as well.
We won't be going down that route in this. Save that for the bloopers.
But we're here today really to talk about environmental monitoring. So in your opinion, what's the aim and the concept behind a well-considered, managed and implemented environmental monitoring plan?
Well, when I look back over the years I've been in this industry, we've always had concerns about how we monitor and control food safety, whether it be chemical, microbiological, or in the last few years, allergen contaminants. The real problem, when I think back to the very early stages of my career, was we didn't have many rapid techniques, in fact, any rapid techniques. My first ever job was as a sound microbiologist in a cannery. We were checking water samples, we were checking sort of can surface samples, so alpine runs, conveyors, things like that. The only thing we had available was traditional micro. So we take a swab, incubate it, see what we got. We may take a dip slide, see what we got. So you were always working behind yourself. Now, if I think about the biggest changes over those 38 years, thanks for reminding me, is the technology that's now available to provide us with that information in a control, real-time manner, so we can actually do something about it. So rather than getting the results a week later and going, we had a problem last Thursday, we can now get technologies which will give us comparable information, not precisely the same, but comparable in an hour or 2 hours. In the case of some of the technologies that we'll talk about today, within seconds or minutes. And that enables us to shift that whole food safety mindset from a we should have done something then to we need to do something now. And that, from a food safety point of view, is immense. So when we're looking at these concepts of environmental monitoring programs, which are required under all the third-party standards, so whether you're producing for Tesco, Sainsbury's, Asda, Morrison's, M&S, or you're just working under a BRCGS standard, they all talk about the fact that you've got to monitor the surfaces, monitor that environment, because if it's in the environment, it can end up in your product. If it ends up in your product, it's likely to hit your consumer. So it's considering the implications of how you employ that environmental monitoring program to give you the information, but the information you want, not going, well, I can test all this, great, I've got this information, what the hell does it tell me? The answer first, what do I want to know? What do I want to have achieved when I've done this? And working back from there to ensure you use the right technology for you to give you that aim and to ensure that the product that leaves your factory or plant is safe. And the consumer can eat it happily without suffering any deleterious effects.
I know that the sort of real-time results thing is something that rings true with me. I mean, working as a site hygiene manager myself, and again arriving at work that evening to see some non-conformance records, results coming through on an email, and like you say, it's from days ago. It's not what's happening now, and having that visibility now to understand what's actually happening within your factory there and then is a massive thing, particularly from a hygiene management point of view, which my background really is. I just, it's unbelievable the steps of, and how far it's actually comes, even since I started in the industry, you know what I mean? Yeah. So I suppose, what are some of the frustrations or the challenges that you would face with current environmental monitoring techniques?
It's about adopting quickly the rapid technologies that are appropriate, and understanding what that technology is telling you. To give you a great example, ATP testing.
Yep.
There's probably very few people that will watch this podcast that don't use ATP or haven't used ATP in some way, shape or form. Great. And as a non-specific hygiene monitoring device, fantastic. I worked with ATP back in the early 90s when the machines were portable if you had a trolley. They weren't this sort of size kit. They were great big lumbering machines that you had to make your reagents up at the time. And you'd get results. Yep. Fantastic. You get relative light units, and it was working out how you translate relative light units to bacteria. And that's part of the problem. As the technology has moved on and gone into handheld, mobile phone sized devices with single-use swabs, how does ATP work? We snap it, put it in the machine and press a button. Yeah, but what's the technology? What's it actually telling you? Great example of that is when people will talk to us and say, okay, well, I've validated my clean for allergens using ATP. No, there's a lot wrong with that sentence. You're looking at the wrong cellular fragment. It's understanding that ATP is a cellular petrol, for want of a better term, cellular fuel. It's in every living cell, whether it's product, whether it's debris, whether it's hands, whether it's food, whether it's bacteria. And if you've rinsed a surface with a 1% solution of a disinfectant, great, it's 1% disinfectant. It's 99% water. If that's got organic debris in it because of dirty lines, and then you go along and do an ATP swab, you're not picking up bugs.
Yeah.
You're picking up a whole range of potential contaminants. So it's understanding, and it goes back to something I was saying earlier, what's the result I want to get? What's the answer I need? And working back from that to say, well, actually, ATP is not the right technology. If I'm looking specifically for bacteria, something like Fresh Check that we've talked about previously, a really useful technology is, a rapid colour change that's specific to bacteria. Utilising that sort of technology. So not regarding, the frustrations can be, to come back to your question, where you've got an environmental monitoring program in place and you ask for a rationale. Why do you test like this? And the worst answer, the most dangerous phrase in the English language, we've always done it this way. Yeah, but there's new technologies about. And if you look at the BRCGS issue 9, food safety standard, it talks about reviewing the technologies you're using to ensure that you are using the latest and most up-to-date and relevant technology for your environmental monitoring program. Because if you're using the right environmental monitoring program technologies, your product is going to be safe. And it's as simple as that. That's what we're trying to achieve. We want safe food leaving the factories so that we can actually have consumers who are capable of coming back to buy your product next week. Because if you've wiped them out, they ain't going to come back.
No. I know it's certainly one of the points I made. I think it was sort of towards the end of one of the soft conference days that we had. We're so quick to embrace new technology in every walk of life, whether it be mobile phones, laptops, TVs, cars. However, there just seems to be that reluctance to come away from the older technologies and embrace these newer ones, which, as you say, if implemented correctly, can give you so much more information and viable and rapid information as well, that, as you say, you can act on quickly and actually physically do something about, which is, we'll keep trying. We'll keep on it.
For micro, if you're dealing with bugs, and as a microbiologist, I always default back to bugs. The gold standard is swabbing and plate count and growing it. Fantastic. It's only in recent years we've realised that actually, unless you're doing both aerobic and anaerobic tests, you're actually biasing and pre-selecting your results
Yeah.
to only give you the aerobes. You know, if you take a sample and put it through a standard plate count route to get the bugs that grow and look at the species that are there, if you put that same sample through a genetic profile, so metagenomics, you get a completely different spectra, an idea of what bacteria are present. Now, it won't tell you if those bacteria are alive or dead, which is a downside to genetic analysis. We'll come on to how we can get around that later. Over a population growth timescale, so you can look at your population flora in the factory over a period of months, you start to get a picture of what bacteria you have. And some of them are weird and wonderful. Some of them cause absolutely no problems whatsoever. Others you think, oh, there's an outlier there, there's something strange. So it's about using all of that technology that's available, using all of the techniques to make sure that what you are getting is an actual complete picture, and not just a blinkered view of what may or may not be present in your factory at any given time. Certainly for micro.
Yeah. So obviously a lot of the technology you're talking there, particularly the sort of genetic side of things, using that in combination with some of your traditional labs, probably. Who would you sort of speak to and to get some information on this and maybe sort of establish your best EMP routine?
We're a good place to start.
We're a good place to start, yeah.
We're good. But there are independent organisations as well. So there's the Camden BRIs of this world. There's Society of Food Hygiene and Technology. There's IFST. Earlier today I was on IFST Food Safety Special Interest Group meeting. One of the things we were talking about was pathogen control. Because obviously that is a key area for that group. So there are individuals out there. You can look at different universities. So National Centre for Food Manufacture, Potter Lincoln, Sheffield Hallam, Cardiff Met, Nottingham. There are universities doing a lot of food safety research who are starting to link up more and more with industry to actually ensure that the output of those analyses and those research projects actually have real world applications. Because it's great having a 58-page scientific paper on something. If it's got no practical real world value, it's not as impactful as it could have been. It's actually considering it, as I was saying earlier, looking at your end result. What do I want to have achieved? Well, actually, I want to have achieved this. Right, that's what I want. Work back. Rather than, here's all this data. What is it telling me? You know, and that's where we're gradually moving towards with environmental monitoring programs.
No, I think it's a big thing. It's sort of a lot of information out there. It's sort of making sure that gets filtered through in a way that, you know, people have got time to understand to digest, understand, have that time to get their head around what's actually happening and what results and it can benefits it actually give to you. So I suppose in a way we've talked a lot about the sort of the environmental monitoring programs themselves and setting them up. Should we have a look at some of the products in isolation?
Look at some of the items.
So one of my favourite ones at the moment, Fresh Check. Do you want to give us a little bit of a...
Yeah, so this is something that has been around for a little while. This is its latest iteration, so it's a single-use swab. It started off as a spray, and it is bacterially specific. So I can, if I have a known hotspot in a factory, I can use this to determine within a matter of 15, 20 seconds if those bacteria within that hotspot are viable. Won't tell me what they are, but it will tell me if by colour change if I have a bacterial presence there. So that will give me an easy indication before I then go on to do any further analysis. It's a very old technology in terms of chromographic changes. The great thing about this particular tech is it's actually made it into a handheld usable form. You know, where I can just take it out, swab my hand. If this goes pink, I'm really worried. You know, and instantly there's a colour change. You know, and straight away I'm saying, okay, well, the hand washing I did before I came in here has obviously worked because, you know, I'm just a sort of a bluey colour, so I'm safe. Good to know. I'm going to make it at the end of the day. And that's great for giving me more appropriate information if I'm in a high-risk factory that I've got bacteria present than an ATP swab will give me. You know, ATP is fantastic, brilliant indiscriminate hygiene monitoring tool, absolutely fantastic. However, it will only tell me if there's ATP, adenosine triphosphate, on the surface. or in the culture I've just swapped. One of the downsides with cleaning disinfection is that we stress the living daylights out of bacteria by trying to kill them for several hours, reduce their ATP levels, and then carry out a test that measures those energy levels. I always liken it to measuring the energy levels in a marathon runner before and after the race. Before the race, going to be high. After the race, really low. Unless you're my daughter when runs past at 25 miles going, hello, and you think, oh, you've just run 25 miles. How can you be that chirpy? And then she collapses, she falls over at the end. So this is more appropriate to me to pick up a bacterial population than an ATP test. Yes, it's analog. However, there is a fantastic app for trending it, for recording that colour as that sample, and then carrying out trend analysis. So combining this with ATP, with traditional micro testing, and with rapid speciation and rapid species testing, particularly pathogen testing we'll come on to later, is a fantastic tool for giving you that information.
Yeah, and it's certainly something, as we just talked about then, it's kind of that, where does it fit? We've got ATP, and people seem to be quite happy that they're getting what they need from ATP, but if you just consider it, like you said, a high-risk factory, that microbiological presence is probably more of an issue for them than just, organic debris or product debris, whatever it is. So to know that, okay, that you're from an organic point of view, debris point of view, that you clean, fantastic. But to also support that with a microbiological result as well. So using ATP and fresh check in tandem, it's like you say, it's closing that loop a little bit more and giving them a little bit more than what they have to then present towards us, when questioned, basically.
Precisely, and certainly looking at the Camden BRI study on that particular technology, there were levels of ATP that were being picked up on surfaces, in the single figures. But Fresh Check was reporting the presence of bacteria. And we knew bacteria is present on the surface because they've been inoculated there. So, levels of 10 to the 3. In one or two instances, particularly for some of the Listeria testing, Listeria mono particularly, it was recording figures of 7, 15, I think 24, which most factories will go, fantastic. But we knew our mono was there at 10 to the 3. Fresh Check was immediately reporting its reaction. So we've got that, we have the technologies coming through, and sticking to we've always done it this way isn't giving you that information that's appropriate to your particular organisation, your particular operation at that time.
No, that's the key thing, that real-time thing. So moving on, I'll have a look at this one here, TBF 300.
The pink spray.
The pink spray.
Again, something else that for many years in the food industry we've talked about biofilms. And it was usually in hushed tones, in quiet corners, because if we did, if we thought we had a biofilm, we think we might have a biofilm. Great. How do we find it? How do we prove it? And when it's there, how do we get rid of it? Certainly, there are methods for identifying it. Got my teeth in gear. Spectroscopy, looking for a slight sort of rainbow sheen to stainless steel, things like that. Benefit with this particular spray is it stains the extracellulose layer, so it actually stains the microbiological element of the biofilm, which most biofilms forms about 80% of the mass, is non-bacterial. Couple that with the fact that most bacteria sitting within a biofilm are actually dormant because they don't have to do a lot because of the structure of the biofilm, which if you imagine a field full of mushrooms, that's pretty much the shape of a biofilm when you get down to microscopy level. So the fluid that flows through carries the nutrients to the bacteria, so they don't have to do a lot. So their ATP levels are low, back to ATP, so the energy levels are down. They're not really respiring, they're not really metabolizing a great deal, so exposing them to a hydrogen peroxide solution to give bubbles by catalase production is potentially giving you a false negative. Staining the polysaccharide layer, now we're talking a bit better, because we're then staining something that doesn't require a biological activity, forms 80% of that biofilm, and gives you an immediate visual representation. That's brilliant because that stays pink. So we apply the spray, give it 5 minutes, wash it off with a bit of fresh water, and if there's pink staining remaining, that's proof positive that we have a biofilm present. And it stays there until you get rid of it. So we've had instances where sites have come to us and you've turned our factory pink. We need to get rid of the pink. No, get rid of the biofilm.
Yeah.
Pink goes with it.
Yeah.
And there are chemicals available to do that, generally based on twin actives, because one of the challenges with removing a biofilm is unless you completely eradicate the biological entities in there, you release it.
Yeah.
And what we have found previously is you decontaminate, remove a biofilm, all you've done is break it up. So your results for a few days afterwards go whoop and peak before they drop back again. Whereas if you're using a twin active disinfectant, one active breaks up the biofilm, the other one mops up. So it's a bit like breaking down the walls of the prison.
Yeah.
Prisons all leg it, but there's people standing there with AK-47s to take them out afterwards. So it's a more effective way of controlling that biofilm.
Yeah. And really good products as well. I've had a lot of experience with the Medasan 327BF myself in the field, and again, utilising it in tandem with the TBF to sort of, like you say, identify the issue and resolve it. And that's essentially what our customers are asking us to do. Find a problem, fix it, and
well, that's it. It's about, as we were saying right at the start of this, it's about harnessing that technological availability to actually achieve what you want to. I want to find a biofilm and remove it. Right, that's my aim.
Yeah.
How do I do that?
Yeah.
Well, I can use this, this, and this.
Yeah.
Fantastic. Right. Now I just need time to do it.
Yeah.
But the best thing with the biofilm is not to let it build up in the first place by cleaning your dark, dank corners, as we talked about earlier, frequently enough that they don't get a biofilm build up. Then you remove the problem.
Yeah. But again, we're back to time frames again, aren't we? We can arrive there at 10 p.m. at night. Within an hour or so, we could have identified something, and within a couple of hours, it's been treated and removed, basically. Yep. Whereas, again, going back to even when I started in the industry, it was, maybe 2, 3, 4 days, or maybe longer, because only after that initial spike that you do get, you're tidying up after that as well. So, yeah, fantastic work. Right, your big moment. Come on.
My loop pixes.
Your loop
what are you on about?
Go and get
well,
them.
One of the things that has really, and anybody who's knows me on watching this will be unsurprised on this. Certainly, one of the biggest challenges, you mentioned about frustrations earlier, for the last 20 odd years, heavily involved in the ready-to-eat food industry, trying to track down where we're getting listeria, and particularly listeria monocytogenes, into products. Horrible bacterium, low number of cases, high mortality rate, anywhere between 18 to 40%, depending on the population. We've had some really well documented outbreaks across the UK and especially in Europe at the moment. The EU member states, it's growing quite rapidly as their consumption of ready-to-eat food grows. Fantastic. So for many years we've crawled over, you and I have crawled over factories together at the wee small hours of the morning, taking swabs, sending them off, and let us know when the results come in, and a week later you go, oh yeah, we had listeria there. We've cleaned 6 times since then, at least. And you change things and you alter things as you go, and then eventually the problem goes away, or you eradicate it and you go, great, what worked?
Yeah.
Something. And one of the things that we've been looking for quite some time is a verification system, so something that will give us the presence of those pathogens in real time, by which I mean I can do something about it. And there's technologies out there, there's phage-based, there's genetic methods that will give us results within a matter of minutes. Unfortunately, every time we've come across them, they've always required pre-incubation. So 24, 36 hours in a controlled temperature, controlled environment before you carry out the 15-minute test, which is great. You're still getting results in 15 minutes, but you've got to wait till tomorrow to get it. If you're dealing with a short shelf life product like sandwiches or sushi, even tomorrow is too long. I want to know if I've got El Mono in my factory now, preferably an hour ago, because I don't, and it's controlling it. These little blue Lupix devices that are genetic based are really the first time I've actually come across something and thought, this works. This gives us the results. We were introduced to it. It's a small company in France who make these. Our French colleague said, we've come across this tech, you might be interested. Yeah, okay, what's the pre-incubation time? Oh, there isn't one. Oh, that's interesting. So we went over to our friends in Spain, to the micro lab. Get me a swab, gave me a stainless steel plate with El Mono on it. I swabbed it, and 90 minutes later, I get a red light to say I've got El Mono present in the sample I've just swabbed. And you stand there, and you look at the red light, and you look at your swab, and you think, where have you been for the last 20 years? You know, you'd have been really helpful at 3 o'clock in the morning climbing over a sandwich factory trying to find listeria, because I could have done something about it there. Yeah. And as we've worked with this over the last sort of 12 to 18 months, and we've now deployed it into customer sites when they've had challenges. You know, I've done a demo at one particular site where we took some swabs in the factory, we went back to the office, and within 90 minutes, I'd got a red light. So I knew that one of those pieces of kit we'd sampled was positive for El Mono. So the technical manager was able to get straight on the phone and say to the production manager, don't start that kit until hygiene had been there. Yep. Now, the challenge with a genetic-based system such as this is the population live or dead? I don't care about dead El Mono. I want dead El Mono. But I want to know if the population is live. And that's where combination technologies come in. So with Fresh Check, I can swab a hotspot. Have I got bacteria? Yes. Fantastic. Right, I've got bacteria. I can then do a Lupix swab, and it'll tell me, is it Listeria? I mean, Listeria mono at the moment. Imminently, we're looking at Listeria at the species level. We've already got Salmonella at the species level. I get a red light, and I get on the phone and say, right, we need to decontaminate. Well, I already know I've got bacteria present. So in the time that it's taken me to do this test, which is about 90 minutes, the hygiene team or production have already been in and decontaminated that hotspot. We then do another Fresh Check swab and say, no more bugs. So I've gone live, viable bacteria, presence of the known pathogen, no more live bacteria in a couple of hours rather than a week. Or if I'm using a rapid file-based system, 36 hours. This way I can control my pathogenic populations and eradicate them there and then. So I can get on the phone and say, that sushi, those sandwiches that we dispatched an hour ago, just put them on hold. We need to remake. I need an intervention into that factory now in order to ensure that the pathogens that are present are eradicated. And I know within Christains and within the wider industry, for those of you that have seen me talk about conferences of these, my Lupix, my precious, watch out for the stuff that's coming out on Valentine's Day, folks, with me and my Lupix, because it genuinely is a technology and a regime that gives me what I've looked for decades, which is control. And that's the thing. Yes, I am a control freak. Yes, if you split me down the middle, it says food safety. Always has, always will. Because I want to make sure that the food that we supply and our customers supply is safe. I've been in that situation. I worked for a short shelf life salad manufacturer for M&S and Sainsbury's for a year. I was hygiene manager to start with. I was then joint hygiene and production manager. And those of you that are in the industry watching this will know that's not exactly the best of normally the best of bedfellows. I used to take myself off and give myself a good talking to every day, because I was responsible for cleaning it and making it dirty again. But the elements there were, this product that's leaving has got to be safe. A pre-packed protein-based salad has maybe a 4 or 5 day at most shelf life. So having a pathogen detection system that tells me El Mono was present in that salad or those protein base 7 to 8 days later is hopeless. There's no point issuing a recall. It's already eaten. By the time you get your recall out or get your results back, your listeriosis complaints are coming in. Something like these technologies that will give me that answer now, that I can do something about it. It's actually starting to put pathogen testing into control rather than management or monitoring. And that to me is hugely exciting. I wish I had another 20 or 30 years in the industry left to know where this is going. With the amount of experience highlights I have, I haven't got long enough left in the industry to see where this will go. you'll be kicking around long after I've departed the industry to do that, as will the young lady on the other side of the camera here, and that's great. I'm actually genuinely jealous of where this tech can go, because if I think about where ATP has gone from and to, when I think where Fresh Check has come from and to, even in 3 or 4 years, from a spray to a swab.
Yeah.
What will be developed next to give us better environmental monitoring and better control over the safety of the food leaving our customers' factories? That's what gets me up in the morning.
Yeah.
That's what gives me a buzz. I know I'm over enthusiastic about it, and that's me. I think one of the things that people have said about me in my career is, "You're too enthusiastic about this." I'll take away the word "too."
Yeah.
The way I am.
Yeah, I know. Well, I mean, you're talking about the technologies moving on, but I mean, even here and now, it's like you say, Peter. It's something we've been after for a long time. It's real time results. It's real time actions, and as you say, real time control on everything. It's just like you say, I'm excited myself to see what the future brings. But
well, the thing is,
for now, I'm actually quite happy that we're here, and
we've got it. We'll work with it.
Yeah.
We'll develop it. It's still got a way to go. It's still an emergent technology. And you mentioned earlier about frustrations, and you mentioned earlier that where are we going? People we rapidly adopt. I mean, we have no choice but to rapidly adopt the developments of mobile phone technology.
Yep.
When I started in the industry, if you wanted to make a phone call, you had to stop, put 2p in the slot, and dial the number.
Yeah.
Now we have a small computer in our pockets all the time. And that advancement of technology, that adoption of new technologies, is something that we're quite conservative in the food industry with a little c, that we don't want to change things because we've always done it that way.
Yeah.
And getting a concept of this is how does it relate to colony forming units in a bacteria? Sort of.
Yeah.
We've learned relative light units for ATP.
Yeah.
We don't always know what they mean. We don't always understand how we got them, but we know what figure we're looking for.
Yeah.
With genetic technology, it's again a different measure.
Yeah.
With this, it's quite simple. It's a red or a green light.
Yeah.
If it's red, you got a problem. If it's green, you haven't. Simple as.
Yeah.
In terms of its detection levels, you have to understand the morphology and the growth characteristics of the microbiological flora you've got. Because if you've just spent 8 hours trying to kill them, gene expression is going to be low.
Yeah.
If you wait till mid-production, I mean, crumbs, Richard, how many times over the years have you and I taken swabs after a hygiene team have finished, it's clear for listeria, 3 hours later, we've got a problem. It's a re-emergent population. So it's not just knowing what the technology is giving you, it's designing your environmental monitoring program to give you those results in a way that is meaningful and realistic, and understanding the time frames, not just of the technology, but also when to deploy that technology. And that to me is the basis of environmental monitoring program. It's not we take some swabs, that's it, we're done.
Yeah.
It's about looking at your whole process, the whole gamut of what's available to you, and going, right, in order to ensure that my food is safe and the integrity is maintained, this is what's appropriate to use. And if it upsets production, sorry, but it's no point producing food to go out of one door if it's then coming back in the other because it's contaminated.
Yeah.
That's just a waste of time.
Yeah.
So environmental monitoring programs are, to me, all about ensuring, with all reasonable precautions, that your food safety management system and safety management program is working adequately.
Yeah.
And protecting consumers.
Yeah. Well, that's what it's all about, isn't it, really?
Absolutely.
It is, and it's a big part that, this senior management buy-in that's mentioned in BRC, it's getting the whole factory, every department to understand that's why we're doing it. It's not just be awkward, not to spend money, not to, delay production in any way. It's there purely to protect the business and produce quality, safe food.
Absolutely.
So lots of good information there, Pete. Lots of interesting products to hopefully go away and discuss. But just as a final thought on EMP, anything you want to finish on?
Just really, in this short podcast, we've done that bit. We scratched the surface. Really is about looking on the web, going to the Chris Daines website. We've got 3 white papers there on environmental monitoring program, on Insight EMP Plus, which is what we're calling the combination of Fresh Check and Lupix. You know, looking at critically at your food safety management program, your environmental monitoring program, and saying, am I using what's best, what's appropriate, and am I getting the information I actually need? Not what I think I want, what do I need? And working back from there.
Yeah.
Always start with the answer in mind, work backwards. Always ED it.
Brilliant. Right, well, thank you so much for your time.
Thank you.
Pleasure as always. Yeah, thank you so much, Pete.
Not a pleasure. Enjoy.
Can you please
Don't
just
remind me.
give yourself, sorry, my bad. give yourself, give
First
yourself.
blooper. First blooper. Yay.
Give yourself,
I don't know if I'm
yeah.
right.
Stop.
Certainly made my, sorry, my house. Right, just keep going. I'm okay, just keep going. Now, sorry, I want to say back to business. But it's back. Maybe we should have thought about it. Okay. And, hello.
Yeah, he keeps kicking me, but that's fine.
Do I? I've
No,
been kicking everyone. Technical. Oh, for fuck's sake, Rich. Fine. Very technically focused episode.
Right,
Right, Darren.
I'm recording.
Last time I felt a bit like I was like round
Yeah,
shouldered.
sit.
It's hard. Yeah, take your time. Can you see all this, Jess, as well? We are going to be going all the way through, so do they need to be closer if you're going to episode? Sorry. And it.
Now you're too far that way. What have you done to your head?
Fancy.
Pause and then.
Fancy.
Yeah. No way. Lord.